Hi everyone! In this video, I'm going to talk about the story of HPV, from infection to immunity. Let me first introduce what HPV is. HPV stands for human papillomavirus. It is a double-stranded DNA virus that infects epithelial cells. Epithelial cells are the cells that line surfaces of our body, such as the skin and the cervix. There are more than 100 different types of HPV. These types are classified based on differences in their genetic sequences. HPV is one of the most common viral infections worldwide, and most infections are harmless and resolve naturally because the immune system clears the virus. However, not all types behave the same way. Low-risk types such as HPV 6 and 11 can cause genital warts. These are uncomfortable but not life-threatening. High-risk types, especially HPV 16 and 18, are strongly associated with cervical cancer. Persistent infection with these high-risk types can lead to abnormal cell growth and eventually cancer. We can also have a look at the structure of HPV. HPV is a non-enveloped virus, meaning it does not have a lipid membrane surrounding it. The outer shell is called the capsid. The major capsid protein is called L1 proteins, which assemble together to form the main structural framework of the virus. There is also a minor capsid protein called L2, which plays a role in viral entry and genome packaging. Inside the capsid is the viral genomic DNA. The viral DNA is associated with histone proteins. Histones are normally host cell proteins that package DNA into chromatin. HPV uses host histones to organize its own genome inside the viral particle. To understand how HPV causes disease, we first need to see how it enters the cells. HPV binds to receptors on epithelial cells. This interaction triggers receptor-mediated endocytosis. This process reflects what we learned in cellular signaling. A molecule binds to a receptor, the receptor changes shape, and downstream processes are activated. After entering the cell, the viral DNA eventually reaches the nucleus. Once HPV DNA enters the nucleus, it follows the central dogma. The viral double-stranded DNA is transcribed by the host cell's RNA polymerase into mRNA. That mRNA is then translated by ribosomes into viral proteins. HPV does not carry its own transcription machinery. It completely relies on the host molecular systems. Two of the important viral proteins produced are E6 and E7. These proteins interfere with normal cell cycle regulation. During persistent infection, especially with high-risk HPV types, the viral DNA can integrate into host genome. This integration often disrupts the viral E2 gene, which normally regulates E6 and E7 expression. When E2 is lost, E6 and E7 are overproduced. E6 promotes degradation of p53, a key tumor suppressor that normally stops the cell cycle when DNA damage occurs. Without p53, cells lose an important checkpoint. E7 binds to the RB protein, releasing the transcription factor E2F, which activates gene required for cell cycle progression. Together, these disruptions remove critical regulatory controls, leading to uncontrolled cell proliferation and increasing the risk of cancer development. Now I want to connect this to our lab. In our bacterial transformation experiment, we worked with plasmids. small circular DNA molecules that replicate independently inside bacteria. We inserted a gene of interest into plasmid, creating recombinant DNA. This plasmid was introduced into competent E. coli cells. Inside the bacteria, the inserted gene was transcribed and translated into a protein. The HPV vaccine uses the exact same molecular principle. Instead of inserting a reported gene, scientists isolate the gene encoding the L1 capsid protein. This gene is inserted into a plasmid vector and introduced into host expression system, usually yeast. The host cells produce large amounts of L1 protein. These proteins spontaneously assemble into virus-like particles, or VLPs. Importantly, these VLPs contain no viral DNA, so they cannot replicate or cause infection. This is recombinant DNA technology applied to real-world medicine. How does the preventive HPV vaccine work inside the body? After injection into the muscle, the virus-like particles, or VLPs, travel to nearby lymph nodes, where immune responses are organized. These VLPs are made from the L1 capsid protein. The L1 proteins are arranged in a highly repetitive structure, meaning the same protein is displayed many times on the surface. This repetitive pattern is important because when identical proteins are repeated closely together, they can bind to multiple B cell receptors at once. This is called receptor cross-linking. When several BCRs are engaged simultaneously, the activation signals become much stronger. This type of activation can occur without the help of T cells. Through this pathway, some B cells quickly differentiate into short-lived plasma cells. Plasma cells produce antibodies, providing early short-term protection. There's also a second pathway. Dendritic cells, which are specialized antigen-presenting cells, take up the vaccine particles. They process the viral proteins and display small fragments on NHC class II molecules. These molecules act like display platforms that show antigen fragments to CD4 positive T cells. CD4 positive T cells are helper T cells. Instead of killing infected cells directly, they coordinate immune responses. Once activated, some CD4 helper T cells become T follicular helper cells, or TFH cells. These cells interact with B cells inside structures called germinal centers in the lymph nodes. Inside germinal centers, two key processes occur. First, affinity maturation. B cells producing stronger binding antibodies are selected and expanded. This improves antibody quality. Second, class switching. B cells switch from producing early LGM antibodies to more effective types such as LGG, which provide longer-lasting protection. As a result, some B cells become long-lived plasma cells that continuously produce high-affinity antibodies for years. At the same time, memory B cells are formed, allowing a rapid response if HPV is encountered again. Together, these mechanisms explain the strong and durable antibody response induced by the HPV vaccine. When a vaccinated person is later exposed to real HPV, pre-existing antibodies bind to the viral capsid and prevent the virus from attaching to epithelial cells. Because HPV must enter epithelial cells to initiate infection, blocking entry prevents infection before it begins. And then is the real-world connection. Cervical cancer remains one of the leading causes of cancer-related deaths among women worldwide, especially in low and middle-income countries. nearly all cervical cancer cases are associated with persistent infection by high-risk HPV types. In other words, this is largely a preventable cancer. In countries where HPV vaccination coverage is high, there are significant reductions in HPV infection rates and cervical cancer incidence. This shows that the vaccine is highly effective. However, disparities remain a major challenge. Access to vaccines, affordability, healthcare infrastructure, and public awareness vary greatly across the regions. Social-economic factors and cultural attitudes toward the vaccination also influence uptake rates. So there is still a long way to go. At the end of this video, I would like to sincerely thank Professor Lee for his devoted teaching and guidance throughout Bio 110. I'm also grateful to my ThinkPair team members for the thoughtful discussions and collaborative learning throughout the semester. Thank you for watching this video.